Yeah,
Here, a
that's
little bit more.
the next. See, the next one is that if I
There's-
open the blinds, is
There's
that
no
better?
spots?
No, that somebody messed around with the lighting.
I mean,
I can adjust it with Kenny, but it might take a few minutes.
Can you open some?
Yes.
I mean, I prefer blinds open and lights off if people aren't going
Okay,
to fall
sure.
asleep. But
Yeah, well, don't. It just means we'll have to check. Oh, no, that's also.
that's okay. That's fine. I like that. This guy never plays on the bass.
You just got one. Oh, no.
It's fine. This is totally fine.
Okay, all right, thank you. Okay, shall we?
Oh, I'm supposed to put this on.
Okay, we'll get started. Welcome, everybody. Thanks for coming. It's my pleasure to introduce Elizabeth Hillman for today's CBS seminar. She was an undergrad and did a master's as well at UCL in physics, and stayed there to do a PhD in optical tomography. She moved to this area to work at a Waltham-based company called Argos. I mean, I call it that. And my hunch is, but correct me if I'm wrong, that it was to develop some non-invasive glucose monitoring.
That is correct, yes.
So that was, and that was maybe relatively short-lived, and she then moved to do a postdoc at MGH, maybe with David Boas.
Correct. Mm-hmm.
And there began her work on neurovascular coupling, about which we'll hear. I'll say a little bit, and then she'll perhaps tell us more. And she, I think, was still doing further work in optical tomography at that point. She got a faculty position at Columbia, where she's now a professor of bioengineering and radiology. And she's done award-winning research, I'd say 3 areas, roughly speaking. One of them is new technologies for optical imaging. For example, fast volumetric brain imaging, like a technique that uses single objective and sweeps the light sheet. And she's also done work on camera-based imaging, wide field imaging of brain activity. So that's one. 2 is computational techniques. A lot of imaging these days involves computation, either for image reconstruction or maybe dimensionality reduction. And then the third area is research agenda in neurovascular coupling, which, as you know, is the basis of the fMRI signal, is important for trying to understand the resting state, and in her case also has been used for studying disease mouse models of diseased brains. So there's going to be some aspect of some of those things, I think, in the lecture today, and I'm
Could
happy
be.
to welcome her.
Thank you very much. We'll see if I can surprise you. So thank you very much for the invitation, and hi. Okay, so apart from these pretty things on the front, I have a financial interest disclosure, which is that the SCAPE microscope that I'll talk about is licensed to Leica Microsystems, and I'm working a little bit with System 1 Biosciences. Okay, so as Kenny said, my lab at Columbia is called the Laboratory for Functional Optical Imaging, and that's because I sort of grew up in physics and I was doing imaging, and I started out in tomography, which is this very complicated sort of scattered light thing, but fortunately, you know, that was around 2001, and things started to really take off in terms of GFP, molecular probes, new fluorophores, quantum dots, all of these cool things that could allow us to put optical sensors onto things and actually image things that would be related to life, like voltage and calcium and the molecular identity of things. And so I sort of focused my efforts onto trying to generate imaging techniques that could kind of grab all of that information out of tissue in some way, whether it was in 3 dimensions or at high speeds. And I sort of really wanted to do things in intact systems, intact tissue, in vivo, so that I could leverage all these different properties, like here, dynamic contrast, so the way in which a dye actually moves around in the body, and leverage that to pull out information, or looking at things like blood flow, here voltage imaging in the heart, and so on. All my videos look a bit choppy here. I don't know if this is a feature of the beaming. And so when I was at Mass General and I was amongst all of these people doing fMRI research, I started to focus the scientific question that I was going to address on this idea of neurovascular coupling, so the relationship between neural activity and blood flow, which, as Kenny mentioned, is the basis of the fMRI bold signal. And I just sort of decided to make that the theme of my lab initially, because it was nice not to just be developing tools, but to sort of be guiding my tool development to address a scientific question. And as a scientific question, as a physicist, this is quite a nice one, because it's kind of a pipes and tubes and relationships over time, and sort of diffusion of oxygen and things that are far less scary than things like studying consciousness. So as I said, I was very, very fortunate to then be alive in this time when suddenly these beautiful indicators came along. And this is one of the first videos of GCamp that was collected in an awake behaving mouse. And so GCamp allows us to express a fluorescent protein inside of neurons that actually causes the neurons to flash in the presence of calcium, which is what happens when neurons fire an action potential. And so at the second part of my talk, I'll talk about how I've kind of gone from my imaging methods that were originally designed to look at blood flow into using GCamp to be able to look at neural activity on this very like macro scale across the surface of the brain. So if I talk fast enough, we'll have a whole section on this at the end. If we don't, we'll do it really quick. And then I'll start my talk by talking a little bit more about high-speed 3D microscopy, because these techniques, in combination with these beautiful genetic techniques and this explosion in all of these nice little buggy thing animal models, allow us to kind of interact with genetics and capture behavior and start to really look at sort of complete neural circuits, with the big problem being that it's really difficult to actually capture the activity and the patterns of things that are going on in these systems unless your microscope can image in 3 dimensions very, very fast. And when I started doing that, that's not what we had. We just had this 2-dimensional imaging in a single plane, which wasn't going to really get us to where we wanted to be in these different organisms. So most of you probably know about how you make a 2-photon microscope image. This is an image of blood vessels and astrocytes in a living mouse brain. And the way you form your image is you scan your spot around and around and around and around as fast as you possibly can and sort of look at each point one by one in turn, right? And so you can make beautiful images, but they take a long time, right? So this took several minutes to take this stack. And so we set out saying, okay, all we have to do is scan faster, right? So let's just keep scanning faster and faster and faster, and then we'll be able to make a really fast 3D image. And then after we sort of built that, we realized we should have done this math first, which is to say that 400 by 400 pixels by 100 depths is 16 million points. And your standard Ti sapphire laser pulses at 80 million pulses per second. And so if you were to just do 1 pulse per pixel, which is the fastest you could go, then you would never be able to image this volume any faster than 5 volumes per second, right? Even if you could scan that fast. And that's only 1 pulse per pixel, which is not a lot of signal. If you were doing 1 micron per voxel, that would be 1/7 millionth of the mouse brain. And you'd be scanning your spot at 200,000 times a second, which is nearly 10 times faster than your average resonance scanner. Okay, so even if you're doing single photon imaging, now you only have 12.5 nanoseconds per pixel to integrate any signal, and the lifetime of GFP is about 5 nanoseconds, right? So this is very difficult. This is like me trying to just look at everybody's face in this room individually, one after the other, over and over and over again. I just have a vanishingly small amount of time to spend on each one of you, right? And so we did try and build this. We built a system that did 100 frames per second down. So this design by Philip Keller and Misha Ahrens tried to then say, okay, we need to implement this in a faster way where we don't have to like shake this fish around. So what we'll do is we'll make the sheet by scanning a laser beam like this. We'll move the sheet backwards and forwards by scanning it in this direction. And then we'll have our objective lens physically moving backwards and forwards to stay aligned with the sheet as it's scanning, right? But that's a lot of moving parts and a lot of complicated synchrony, so they never got to faster than about 1 volume per second here. Plus, Fishy here was like stuck like this, which he didn't really like, and you can see all of this distance that you need for these physical objective lenses to all be around each other, and so that imposes limits on your numerical aperture and your immersion medium and so on. So we could see, though, that it was going to be helpful to use a light sheet approach. We just said, can we get these benefits but not have to translate the objective or the sample? And maybe we can actually get rid of these lenses and use a single lens. So we came up with this geometry where we basically illuminate the sample on an angle from above, and then we detect the light back through the same objective lens. Okay? So we can still get that near 90-degree crossing angle between our excitation and emission, so we can still get a pretty good point spread function here. But one of the things you can immediately see is now you don't need to have that sample floating in space. It can be an intact sample here, which can be my finger or a mouse brain or whatever you like. We can now use water immersion and high numerical aperture objective lenses, so we don't have to worry about that physical distance restricting what we can do from needing to illuminate from the side. And I'll explain to you in a minute that we do actually indeed like focus our camera on this oblique plane, which is kind of weird. People don't think that you can use objective lenses that way. I didn't know that, so we just did it. Anyway, and so then instead of bouncing the objective lens up and down, we came up with a way of basically moving the sheet from side to side. So you get this sort of rhomboid-shaped 3D field of view, but it's a really nice benefit that nothing physically has to move at the sample. So I'll explain kind of quickly how we do that. So I'll start with a quick explanation of how confocal microscopy works. So this is a confocal microscope. So you have light coming in here, goes down to your sample and illuminates this point down here, and then the green light comes back, goes over here, and forms another focus here, right? And so the key thing about confocal microscopy is when you move this mirror backwards and forwards, you see it moves that spot down here. But when it's in its moved position, the light coming back is bouncing off the galvo that's in the moved position. Okay, so it's still going to come here, and it's still going to create a stationary spot here. But that spot up here is going to correspond to the location that the spot is moving around down there. It's like me having a mirror here and moving the mirror, so the thing's going to go like that, but the thing I'm going to see in the mirror is going to always be the spot. So for confocal microscopy, all I have to do is put a pinhole here and a single detector. I scan my spot around, collect the signal over time, rearrange it, and put it back, and it's an image. Okay, so that's called confocal scanning and descanning, all being done by that same mirror. So this is what our current version of SCAPE looks like. So we make a low numerical aperture beam here, not a fat one, and we put a cylindrical lens up here which preconditions the beam. So now it comes off and goes into the objective lens at the edge, which causes it to come out like this. And because of this cylindrical lens, this is now a sheet, right? So we've created this oblique sheet in the sample. So I can look at the light that's coming back, and that's going to actually form here a nice three-dimensional image of that oblique plane, right? Now this is a strange feature. This is basically similar to remote focusing. It's mapping this full three-dimensional image plane, object plane here to this three-dimensional image here, and this is called perfect 3D imaging. And this is where it surprises people that you can use objective lenses like this, but essentially everything that gets scrambled here gets unscrambled here and put back where it should be there. So if I want to then take a camera, I can focus onto that plane, and as far as the system knows, I'm down here focusing on the plane right here, okay? So I can create a focused image onto that oblique plane. So the cool part comes now with this galvo mirror. If I now move that galvo mirror back and forth, I can cause this light sheet to move from side to side down here, right, which is what I want. We don't change the angle. We image the galvo onto the back focal plane of the objective lens. But as you can see, we get that same effect that you get in confocal microscopy. So even when the mirror is in the moved position, the light gets corrected on the way back, so this stays stationary. So no matter where I move this mirror and wherever I move that plane, the camera stays focused on that plane. So instead of having to worry about independently moving the light sheet and the camera up and down at the same time, I achieve both of them moving at the same time by just using one single mirror. So I can move that mirror wherever I want, wherever it is, I'm going to see a focused image of that plane. So that means I can do just 20 lines per second on this Galvo and collect data at 20 volumes per second. So I don't need a resonance scanner or anything complicated. The other thing that we do is we actually only read out as many rows on the camera as we need depths into the sample, because each image that we're taking is actually a depth section into an oblique depth section into the sample. And so I can read out a standard CMOS camera at about 2,000 frames a second if I'm only imaging 100 rows. And so I can use those 2,000 frames per second to spread them out along that volume, right? And because I'm only moving this, this is not rate limiting at that kind of speed. But I just grab the data out of the camera as fast as I can, stack those images back together, and I get a volume. We can put an image splitter here, which puts two colors side by side, and that's really helpful when you want to do dynamic imaging of multiple indicators at the same time. So the data coming out of the system looks like this. It's these sort of sequence of oblique planes. This is really slowed down. But then I just stack them together, and now I have a three-dimensional volume. And a really important feature here is that there's no image reconstruction. This is optical sectioning. This is really taking multiple planes, and then you stack them back together rather than needing to do some sort of projection computation. And that's really important because this data is huge, absolutely huge. You have multiple volumes coming out per second. You don't need to be doing computation on all of those. Another really nice thing here is then there's no movement here at all. This is not moving. You don't need to physically be holding onto your sample in order to move it. And so then you have the opportunity to do things like, say, photostimulation down that same objective lens because it's not bouncing around, okay? So here's a little movie of what the system looks like in action. So this is images of the zebrafish brain like coming out just as the sheet's moving backwards and forwards. Another nice thing is, oh, there's the fish just sitting in a dish under, this is a standard 20x 1.0 NA water immersion objective lens with a 2mm working distance. That's the data, there's Venkata. It's a nice thing we don't have to come through the eye of the fish here as well. We can do this from above. So we were collecting data here for an hour on this fish while it was having febrile seizures, and we get 1.5 terabytes per hour just sitting there, which is what he was doing. He was like, "Why are you? What are you doing?" And then yeah. Okay. So this was really Venkata's PhD project, was to take our original design, which actually used this weird polygon mirror, and turn it into something here that's super stable. This is actually a system we packed into a suitcase and brought to Germany last summer and ran at a course in Dresden. And so it's a really simple layout. It's a simple little laser here. There's one Galvo mirror here. This is the primary objective lens, and this is where the sample goes. And then this is the image rotation piece, and then this is the color image splitter and the camera up here. One cute feature is that actually we can rotate the objective lens to any angle, and we can image at any angle, because again, it's just a mirror, and what happens forwards goes the same backwards. So a lot of people like to use it inverted or upright or even, to the side. Okay, so what can we image? I've convinced you, maybe, that it works. So some of our, this slide's always crazy. Here we go. Lots of stuff. I'm not going to talk about all of them. This is this weird Volvox thing that spins. We were able to actually image it while it spun. We imaged tardigrades at Cold Spring Harbor last year because tardigrades are awesome. And I'll give you a little bit of a review of some of these other things that we've done. So we focused our attention on a lot of different aspects of kind of continuing developing Escape, making it higher resolution, deeper, different applications. And then we engaged in several kind of like intense collaborations with people who would really benefit from using it. And so Wes Gruber here at Columbia, he studies the Drosophila larva, and he studies the motor and the proprioceptive system of the Drosophila larva. And so when I met him, he was trying really hard to understand the role of these different proprioceptive neurons along the body wall of the Drosophila. And he wanted to understand, how they encoded the position of the larva, but poor Rebecca was stuck kind of trying to squash the larva between two pieces of glass and sort of put them on ice and have them kind of squirm while she was imaging a single plane using confocal. And understandably, the reviewers were like, well, I don't know, is it really crawling? So we came up with a way of actually just letting the larva crawl and then just imaging it as it was doing what it was doing. And so this is just looking at GFP, but these are those dorsal proprioceptive neurons as the larva is crawling. This is the top view from above, and this side view. This is in real time. This is imaged at 10 volumes per second as the larva is just crawling along. And then this is the view from below. This is the ventral nerve cord. These are sort of the projections from that ventral nerve cord. And these are those VPDA proprioceptive neurons. And from the side here, you can actually see that they kind of like flip up into the body wall as the larva is crawling. And so this gave us just our first sort of structural, what happens to the actual shapes of these neurons. And Wes's research was really involved in what were sort of the molecular cues that were patterning the development of these dendrites into these particular positions. And so here we're understanding what's the function of them having those particular patterns. One thing that's nice about this 10 volumes per second is it turns your imaging into capturing the motion, and it doesn't really suffer from the motion artifact. So you can take single frames of this 3D volume and actually trace the different positions of the dendrites as the larva is crawling. But the really cool part was then when we could co-express a static marker like TG tomato with an active marker like GCAMP. So this allows us now to not just look at the way that those cells are deforming, but to look at then how they're firing in relation to the way that they're deforming. And so now you can see as the segments compress, the little dendrites get brighter. This little thing is an axon that's now flashing in sequence. And the individual cell bodies as well are actually changing their brightness, right? So this was a lot of work by my student, Wenzi, in collaboration with Rebecca from Wes's lab. And so what we have to do then is take that data, we have to track those cells in time. So tracking those cells now gives us the behavior, the position of those neurons, the distance between the different segments, the acceleration, velocity of those cells. But of course, once we've tracked them, we can now also extract the signals from those cells and look at the GCAMP. We ratio the GCAMP signal to the red to make sure that we're not getting any artifacts from the red just simply moving around or being compressed. And then when we've done all of that, we can basically go through every single different neuron that we have in that proprioceptive system and characterize when and how it fires in relation to when the segments get compressed. So we got these time courses and we were like, that's kind of not as interesting as we thought it was. I mean, you know, it's cool, but like it's not as cool as the movies. But anyway, so we were like, that's all right, so they fire at different times. But then we were like, okay, well, what if we just like line them up? And so this is a little bit more interesting. And what we found was that each of these different types of proprioceptive neurons is kind of responsible for monitoring a different piece of the crawling, right? And so people had been studying these and like knocking them out. You knock one out and the larva crawls a little bit more slowly, right? But what this tells you is that each of those different neurons is particularly positioned to either be a stretch sensor or compression sensor and to report back on its little piece of the movement that's happening. So if you knock one of them out, it's like being a little bit drunk, right? And you just like walk a little bit more slowly because you're not quite getting all the feedback that you're used to getting. But they work together in concert to report all these little bits of the information as some sort of continuum. What that also let us do was sort of come up with this idea then that, you know, the signal of those cells was now a function of, in this pair, the compression between the two neurons. And so I developed a little geometric model of this, thinking, okay, if this is like two rigidly coupled sort of bars here, I can actually reduce the dimension of this movement essentially to turning and to compression. If you look, he's not just turning like this, which is what we hoped. He's kind of bobbing and turning. But if I can characterize that, I can predict the movement from the cells. And so I was really pleased with this, even though it's kind of nuts. This is the original data. This is my model. And this is now just predicting its motion from the firing patterns of those two cells. And again, he looks a little drunk, but it's pretty good, right? I can really predict his position in 3D space just from those firing patterns. And so we were quite pleased. Essentially, it sort of suggests that the difference between these cells is telling it about turning, and the sum of the cells is telling it about scrunching. Okay, and so that's continuing, and now we're applying that to a whole bunch of different aspects of the nervous system, because essentially we can image all of the neurons in that nervous system and start to relate their patterns to behavior. Another project that I haven't really spoken too much about until quite recently, because we just recently posted this on the archive, is our work looking at olfactory epithelium, which is a collaboration with Stuart Firestein at Columbia and Wenzi in my lab, and actually his wife, Lou. And so what they realized is that they could use SCAPE to be able to image the olfactory epithelium inside of this really gross, like hemi-head mouse prep, which is an in vitro living prep, which we can perfuse, which allows us to actually flow odors into the nose in the way that an odor would normally go in. But we can image these curved turbinates of the olfactory epithelium at about 6 volumes per second over about a millimeter by millimeter field of view here. And so from the side, we can actually see details of these individual cells and these little knobs. And so by flowing different odors over the top of this, and by doing a ton of analysis to pick out all of the little responses from individual cells, we can characterize the response of every single cell in here to different kinds of odors and different kinds of mixtures. So this is a way of doing essentially very, very high content profiling of the properties of all of these cells. So this is sort of spontaneous activity, and then you'll see this wave coming over as an odor comes over. So we're picking out each individual cell, and then for every cell, we can get the profile of the way that that cell responds to an individual odor or a mixture of odors, and we can do that in tens of thousands of cells within one individual prep. And so that allowed us to do this kind of really intensive sort of analysis of the way that these different cells were responding. And our sort of big result, which actually agrees with work, theoretical work, is that it would appear that there are some cells like this one, which actually, although it has a big response to a particular odor, when it's given that odor within a mixture of another odor, it actually has less of a response than if it were getting that odor alone, which is suggestive of there being like an inhibitory and also an enhancing effect of different odors, which suggests that the encoding of mixtures of odors, even at the peripheral organ in the olfactory sensory neurons, is being sort of modified and modulated, and the messages getting sent to the brain aren't necessarily just a pure linear code. So that was kind of fun. All right, and another study, so this just came out on Friday, and it's not really a study, but we were kind of dared by the C. elegans people to have a go at imaging C. elegans, so we did. And it took a lot of improvements to the system to really maximize the resolution, to really maximize the efficiency of the light. So the big problem that we understood from the worm guys was that in spinning disk confocal, you have a limited number of depth rows, and you also have a problem with photobleaching. So this looks kind of twitchy, but this is actually 5 times sped up because this was imaged for 5 minutes and showed really very little photobleaching during the course of those 5 minutes, despite having a much denser sampling there than you would get with spinning disk confocal. And so that allowed us, oh, by the way, and one of the nice things about our system is we can change the numerical aperture of the light sheet. And so for samples where we want to do, it's not going to work, is it? Okay, for samples where we want to do a long distance, we can use a lower NA light sheet. For the worm, we can use a higher NA light sheet over a narrow depth of field. So here's a little photobleaching comparison that we did with spinning disk. So this is going up to about 2,300 volumes, and you can see this worm basically turns on its side and dies after about 2,600 frames, whereas we've lost very little signal in the SCAPE imaging, matching the same exact signal-to-noise sampling rate and so on. And so we did some pretty nice analysis, I think, here that's shown in the paper, and I'll sort of refer to this again later on maybe. We cheated here. This is a fairly immobilized worm. There's a lot to still be done on doing really good tracking of the moving worm. But we extracted signals here from 113 cells within the head of the worm, and we looked at the correlation coefficients between these signals that we got. These are just raw GCAMP signals coming from the worm. And we did this kind of nice analysis where instead of looking at the correlations during the entire 10-minute run, we looked at correlations and how they changed as a function of time. And so we actually were able to cluster these correlation structures into these different clusters and look at how these different kind of state dynamics seem to be switching. And again, if I get to it, I'll talk about this a bit later on, but essentially my first approach here was like, okay, we've got now this neural signal, let's look at functional connectivity in relation to the connectome, which is the physical connectivity. But what we're basically seeing is that apparent functional connectivity is changing over time. So there's different sort of states in which neurons are correlated to each other, and those actually go through these fairly predictable transitions in this anesthetized worm. But then we got this super fancy camera. So all of this was done with our original system, but this camera can do 1,000 frames per second full chip, which is about 10 times faster than our original camera. And so by now having 1.3 gigahertz pixel rate, remember 2-photon is limited to 80 megahertz, so this is like a lot more. Then instead of having this field of view, we could extend it out to a much larger field of view. So now this is imaging at over 1.3 gigahertz over this like much bigger field of view, and so now we can actually let the worm crawl around while still sampling the volume with enough spatial resolution to see single cells. So this is our current kind of favorite worm. So this is a GCamp tag RFP worm. He's trapped inside of a little arena, but we're basically now doing 26 volumes per second imaging as he's crawling around. And that 26 volumes per second makes all the difference when you want to then be able to track one cell from one frame to the next in 3 dimensions. So if we can just solve the issue of being able to track hundreds of cells in massive, ginormous 3-dimensional data sets, we're pretty much ready to now try to decode what's going on in the worm, kind of. We couldn't help but apply this method to zebrafish heart imaging, which was just sitting there as a really exciting application as well. This video is slowed down because it was acquired at 100 volumes per second, so this is shown at 1/3 real speed. And you can track every single red blood cell going through the heart as it beats. And then we can do the same in GCamp heart here and look at the way that the calcium waves are moving across the heart. And just to give you some idea of the parameters here, we're imaging nearly 13,000 frames per second, but we're only using about 1 milliwatt of the sample. So that big integration time benefit that I talked about ages ago is coming through here on basically allowing us to image these in vivo processes at these really, really fast speeds in real time. Most people that are imaging these samples are kind of doing one plane at a time and like gating, right? But this allows us to look at things like arrhythmias, and it's 400 times faster than resonance scanning confocal. So we've been working with Kamara, who's a pediatric cardiologist, to now quantify information from these, to look at abnormalities in the development of this system. We've been doing a lot of tracking technology here, so working with a group in Japan doing a deep learning-based tracking of these cells. And that allows us to look at their trajectories as well as looking at now the calcium signaling as a function of the trajectories of these cells. Another application that we realized could be quite useful would be the awake fly brain. And a lot of people want to image these flies, walking on a ball and sniffing odors and stuff, but they're not really very amenable to your standard light sheet because they can't really be held in agar and such. And so this was one of the first images that we got with the mushroom body here and a pan-neuronal GCamp. This is the entire brain. These are the eyes, and we could image this field of view at 10 volumes per second. So that got Richard's attention there, and so Evan and Neely in his lab and then Wenzi have been working really hard on this. And so we've improved the imaging to the point where this is now the whole fly brain. We can do dual color, so we can actually label specific types of cells with a different color. I'm a big fan of like the pan-neuronal or cytosolic GCamp, and then basically try to see everything that's going on and then use the second label to kind of pick out specific circuits or specific cell types that we want to be able to record from. And then we can do this real-time imaging. This is the view from above and the view from the side. This one's kind of boring. But you can still apply things like CNMF to this, do your dimensionality reduction, and find numerous different types of cells. And this is real time. So this isn't an averaged response. This is real time firing patterns of these neurons when you present one odor, when you present a different odor, or my favorite, the ones that don't care what odor you're giving them, they're just bapping away doing something else, right? And we can do all of this in the context of recording the behavior of the fly on the ball. And I'll talk about this a little bit later, but this is sort of really, with Richard, we've been thinking a lot about brain state, right? Thinking about how a fly might be changing its response to an input from moment to moment. And so now, instead of having to think about our experiments where we present something over and over and over and over again and then just get the average response to that thing, now we can start to look at sort of like what the actual background ongoing activity is, what the state of the brain is in a particular moment when we present something new, and whether we can use that, for example, to predict the way in which the fly is going to behaviorally respond. We've been working on 2-photon versions and dual labeling now, looking at, say, nuclear labeled and cytosolic, and this is a weird representation here where we've done a spatiotemporal unmixing to characterize kind of different dynamic circuits as different colors, and here essentially we're presenting, this takes a while, 3 odors in succession. So you'll see an odor response And so we can basically get the activity in such a way as we can really look at all the different regions and the way in which those odors actually emerge within the whole brain and then the response of the brain afterwards. This is all a bit slow. I'll give you the next odor and then, come on, 43. Oh. Yeah, so this is sort of a summary which is a bit intense, but like this is one odor, this is a different odor, and this is another odor again. And so we can look at all the full sequence of like where that representation of that odor comes through the brain at what point in time. And both of these odors here, I'm really excited to see, have this weird sort of aftershot, very matched pattern in their response, whereas the other odor has a very different latency. So that's work that's sort of in progress. I had to throw a zebrafish in here because I'm sure that there's some zebrafish people here. This is our Ghetto zebrafish visual stimulation system. Just to prove a point, that is very easy to do this stuff. Here's a nuclear localized fish, just showing that we get this really nice resolution throughout the brain. You'll see what's nice is even if the fish kind of twitches, as long as you can sort of computationally fix that afterwards, you don't need to worry too much about that motion. And so you can do depth color coding here to show these single neuron responses. We've also put together a 2-photon version that now can allow us to see visual responses in these animals. This is a less ghetto version where we used a projector, and here we actually have another camera that's monitoring the behavior of the fish at the same time. So you can build up some really nice, complicated behavioral paradigms here. This was something we did at Cold Spring Harbor, which is basically a drinking game where, like, when the fish actually flicked his tail in the right direction, we drank. It was like 2 in the morning. But I do believe there are some serious zebrafish people doing this now. We've shared the system with quite a few labs. Okay, so we did mouse brain on a dare originally for the first paper, but it turned out to work really well. So for my own work looking at the vasculature, this is allowing us to do 10 volume per second imaging of the vasculature of the mouse cortex. And this is during just single trial during a stimulation of the whisker. And so this allows me to select different branches of different vessels in a vein, and you can see the dynamics of the dilations of those vessels in this 3-dimensional network. And it is a 3-dimensional network, which is why it's really important for us to start to understand the dynamics of the vasculature at this scale, instead of looking at one branch at a time where you then can't really infer what's happening to all of these different resistances. This data also won us a grant to study the acute response of tumors to radiation therapy, because this is a big area for understanding how cells hone to tumors and how blood flow is affected by different cancer therapies. We can also do this, which actually means we can get this 3D imaging as we scroll around with the microscope. And so this has prompted us now to be developing a medical device that would hopefully allow us to guide surgery intraoperatively, because we can get this very high-speed, stitchable volumetric imaging in vivo. Of course, you all are interested in neurons, so we also do neurons. These are apical dendrites of layer 5 neurons in the mouse that we're imaging with Randy Bruno and Sam, a postdoc in our labs. So you can see we can capture spontaneous firing in these apical dendrites over a 1 millimeter field of view without needing to, like trace individual places or do random access. So this is 488 nanometer excitation, which is crazy because it's in the mouse brain. And this is at about 150 megahertz pixel rate. So I can take that data and do dimensionality reduction, which is important for us to sort of turn that from a thing that looks like lightning strikes into something where we can actually interpret that multiple neurons were firing, right? Now this was a fairly simplistic dimensionality reduction. So essentially what we did was we found these firing patterns that were present in the data, and then we classified every pixel that had that firing pattern as belonging to that particular dendrite, right? There is a lot more dimensionality to this, so we sort of grouped together cells to get these pretty nice sort of complete dendrite representations. And mathematically, what this looks like is basically now you've got these spatial components and these temporal components, and you're assuming that you can recompose your data by multiplying them back together, okay? Okay. So I'll show you this movie, which is now going to be those colored classified dendrites turning on and off. What's nice about this representation is that essentially what I'm now saying is that each time one of those neurons is firing, it's going to flash. So I can also assign them a musical note and wake you all up. And so we chose the musical notes according to when they fired first. So they're going to go in an ascending scale according to when they fire. Like 3 minutes of this, so you'll have to wait for the album. And data is like so boring when you don't have music after that. So we're also building a 2-photon version, which should get us deeper and better sectioning. So that was all single photon. This is the same mouse actually without music, getting down to about 350 microns. This is old data, but Hung in my lab has been working really, really hard on this. And now we're getting to the point where we can go up to about 30 volumes per second with near uniform sampling using 2-photon excitation. So that's not published yet, but that's coming quite soon. And then I just threw a few things in here. When we were in Dresden, we had the opportunity to image these organoid chips. And I got really excited because we were actually able to image within the actual PDMS chip. And so I really like this as a way to think about not just looking at these samples sort of longitudinally and intact, but we can do things like look at when we flow a drug through, what happens to the calcium signaling in different layers of these 3-dimensional cultures. So we're really interested in extending in this direction because there's all this growth in the organoid field and in the sort of human-derived sort of drug development space. Another area that we were playing around with is looking at cleared tissues. So this is half of a brain, a 400 micron thick volume that we were able to image in about 230 seconds. So just realizing that we can image really quickly samples that currently are taking a really, really long time on confocal microscopes. And we were really pleased with this resolution. It's actually sufficient to trace all of these fine processes using our system. We've done retina as well. I didn't put retina in because I knew I'd be running out of time. But this is an expansion microscopy sample here that's 4 millimeters long, 1 millimeter wide, and 400 microns deep. And we imaged this whole thing in 8 seconds. Which is kind of important for these samples because they tend to sort of swell and wobble and you don't really want to take a long time to image them. And this is a piece of spinal cord that we included in our recent paper that was expanded as well. So it's quite an effective way of imaging that. And this one we actually imaged 2 millimeters deep into the sample using our objective. We're sharing the system. We've been putting a lot of effort into bringing it to courses, and now we're helping people to build this kind of simplified version for themselves, and we're also working with Leica to commercialize it. Okay, so I think I have about 10 minutes left for part 2. Are we all okay? Do you need to stretch? Okay, let's talk about something else. So that was SCAPE and trying to just give you an idea of some of the more intense projects that we're doing in the development. So the other side of my work now is thinking much more about whole brain. And it's sort of funny because I've had this sort of unique view of whole brain now in these little bugs, but now we're thinking about whole brain on the scale of the mouse. And the system is super simple. It's just LEDs and a camera. But I can now leverage the power of GCamp animals or other fluorescent indicator animals so that I can put my blue light on and get green light back through a filter. And now I can be measuring neural activity. Historically, we did what was called intrinsic signal imaging, which is basically using 2 different colors, green and red, to look at hemoglobin absorption. So we're looking at the effects of blood flow changes in the brain. But by combining these 3 things together, we can look at neural activity and blood flow all at the same time by just basically strobing and collecting these images in sequence. One thing that's really, really important, a lot of people are now starting to do this kind of imaging, wide field. But you can see there's a big overlap in the absorption properties of hemoglobin and the excitation and emission of the GCamp, right? And so you have to be really careful. If you just take a raw fluorescence image, it's going to have these weird vessel artifacts over it. And it's actually going to have dynamics that correspond to the blood flow changes that are actually correlated to the neural activity. And if you don't correct for that, you're going to get all kinds of weird-looking things that might look like inhibition, and they're simply not. So you can't just mask out the vessels. That's not going to solve your problem. So by simultaneously measuring the hemodynamics at the same time, we can actually correct for the presence of the blood flow changes and get back to a signal that looks much more sensible and doesn't have those artifacts, and therefore actually corresponds much better to the electrophysiological signals. And we've done validation of that. But this is, it's a little bit of a, it's not a dark art, but it merits looking at the theory of like why there's crosstalk and how to correct for it. So we've actually written some papers just to sort of try to guide people on that. And so anyway, we advanced our prep from doing like a small window to doing the full dorsal surface of the cortex. We thin the skull, coat it with super glue to prevent regrowth and to make it more transparent. And then we laser cut these little plastic antlers for the mice to wear. And then we can keep those mice for a long time, and they're quite happy walking around with these little plastic head plates. But when we want to image them, we can clip them into our system and allow them to run on usually a horizontal wheel while we're imaging head fixed. And as I said, this is from the Allen Brain Atlas, but it shows you that this dorsal surface projection of the brain really includes a whole bunch of really nice sensory regions, auditory, visual, retrosplenial, and motor here. So I can do something really simple just to sort of prove that this is okay. I can flick the animal's whisker 50 times and do an average, and I'll show you the neural response here, and this is also the blood flow response, total hemoglobin, oxy, and deoxy. So there's the neural response in the whisker barrel, and then there's that blood flow response. And have we got any fMRI people here? All right, well, if you were an fMRI person, you would be like, thank goodness, because it really shows that there really is a nicely coupled, localized blood flow response, which is what you would be getting your bold signal from. Okay, but you also saw some kind of interesting things here, like there's a neural response here, but there's actually also a response here, and there's a little bit of a response here, right? So there's all this stuff going on throughout the cortex that's happening at the same time that we don't tend to look for when we're just looking where we're expecting to see a signal. So that should convince you that it works. And we've applied this in a lot of different ways in the lab. I've done some studies looking at, for example, endothelial propagation of vasodilation, knocking out endothelial propagation, finding, I'll happily talk about those papers, but essentially realizing that that blood flow modulation is dependent on endothelial health, and therefore cardiovascular diseases, diabetes, and drugs that impact endothelial function will actually be probably having an effect on your brain as well as your cardiovascular system. We've also applied this for looking at postnatal development, because early bold fMRI studies actually showed what looked like an almost inverted response in the newborn brain. And everyone was like, huh, that's weird. So we studied it. Now it's going to do this thing where it disappears. Okay, you can look at the papers. We studied it, and we found that actually postnatally, this neurovascular system actually develops and does go from being initially inverted to then coupling up and being positive. But the thing I'm going to talk about more today is that who here is at least aware of resting state fMRI? Okay, who hates it? Yeah, that's what I thought. So I got on this, about 10 years ago, I wrote like my second R01, I'm like, I'm gonna prove that resting state fMRI is garbage, right? Because how could it possibly be? You've got all these correlations and it's probably just an artifact. So I'm here, I've got these methods should allow me to like look at what's going on in the vasculature and maybe in the neural activity, and maybe I can stop the NIH from wasting all their money on all these studies looking at resting state fMRI. And as you all know, resting state fMRI works by basically taking a bunch of scans as the person's just sitting there, and then picking out a region and finding that actually there's all these other regions at other places in the brain that seem to be correlated to that region. And then we infer that that region is functionally connected, right? And so actually, for the last 10 years, this field has been describing the human brain as this very elegant, complex, interconnected a network of all of these different correlated regions. So we said, okay, we've got this mouse, we do electrophysiology, we do calcium imaging, doing nothing, this animal has this nice activity. What does that activity look like if we zoom out and look at the whole brain all at once? And it looks like this. Or it looked like this. This is an older data set, but this is basically the first one where Sharon was like, you have to see this, because the neurons are doing this weird thing with these correlated patterns all throughout the brain. And we know that this is neurons because this was a Thy1 GCAMP6 mouse, which was expressing GCAMP only in its excitatory neurons of layers 2, 3, and 5. And we've corrected for hemodynamics. So these patterns were really bouncing around, and they were real. And so we looked at the blood, because that was where the fMRI bold signal should be coming from. And it doesn't look quite the same, right? It looks kind of not the same. But then the relationship that should be between the neurons and the blood flow is a convolution with a hemodynamic response function, which is basically slowing down the signal and smoothing it over time. So if we now estimate the hemodynamic response function, we can now actually predict these hemodynamics from these neural signals really, really nicely. Okay, so we published this and said, you know, guys, actually, perhaps those patterns that you're picking up with your fMRI are really being driven by these patterns of neural activity. But we hadn't really seen these before because we were averaging and averaging and averaging and getting rid of them and calling them noise. And even when we looked at them on one side of the brain, we were calling them noise because they just looked like random flashes, right? But when we went to this bilateral view, we could see this bilateral synchrony that really made it clear to us that they weren't just ordinary noise. And so what are they? So you can k-means cluster those patterns, and they create these maps, which look really cool, and so suggest that they're fairly similar to these topographic maps that we've been looking at in our atlases. And you can compare these to what other people have measured as resting state networks in the hemodynamics in rats and mice, and also to the human brain. So we really feel like these patterns are somehow depicting the signals that are being picked up in resting state fMRI. And one reason to care about that is that actually resting state fMRI, as much as you might hate it, one of its sort of really interesting features is it's been able to differentiate between different populations, right? The schizophrenics versus a healthy brain. It doesn't really work on the scale of an individual, it works on the scale of like hundreds of patients, but there's something different about those brains that's popping out in the features of these resting state functional connectivity networks. So if we can understand what the neural basis of those are and what it means when they appear to have a different functional connectivity network, maybe we'll be able to make those results in fMRI a bit more actionable and relate them either to the mechanism of disease or turn them into a more robust biomarker for disease. So we've done a lot of things here. As I already said, I'm a big fan of dimensionality reduction. So this is a sort of color merge of that data over time. I can pick different regions. I can find those regions over time using that same method as I showed you with the dendrites. And sure enough, if I just pick these regions on one side, it finds all these other correlated regions on both sides. It pops that out really nicely. I spent some time with the Allen Brain Atlas actually picking these regions and looking at whether they actually correlated well to the spreading of viral injections. And you know, they look kind of like topographic regions. And so essentially what I'm saying here is I can simplify what looks like those complex waves as basically these regions all kind of like turning on and off at different times, which helps me get a little bit of better grasp on what they might mean, whether there's motifs, sequences, patterns in this data that somehow might relate to something important. And so if I take my original data and reform it just using those 9 components, I can recapitulate quite a lot of that information, which is pretty nice, right? I can also turn it into music. So my student Nick was like, neurons are piano and blood is a violin. So I'll just play it because we're getting low on time. You can sort of watch the video instead of needing to. Now he's going to sit there for a while and play a little tune just while he's sitting there, which is cool. Okay. So you can give him ketamine, and he does this, which is fun. So obviously, we needed better behavior, right? And of course, beautiful deep lab cut came along, so we got these beautiful cameras. We can see the underside of the mouse now, the side, the front. Now we're going to add another mirror, see both whiskers, both eyes. Now we know we've got this cortical representation of all of this stuff, then now we really need a much better representation of the behavior of the animal, right? And so we also found a better mouse. So we're using this Taiwan jail gecko, which seems to express better in layer 4 and somehow gives us a faster and much more detailed representation. So now my clusters have these amazing regions of differentiation here that's now got me talking a lot to the Atlas people and saying, well, maybe we should be using these functional dynamics as a way to differentiate between different regions rather than just basing our assessments on anatomical stuff. So these are just being differentiated based on the different temporal properties of this activity. And this is starting to look awfully a lot like the Human Connectome project. But the nice thing is now we can really get these representations of every single one of these regions as different time courses while we also have that behavioral data. And so this is sort of where we're at now. We've got the ability to now extract a lot of behavior, to dimensionality reduce all of this. And of course, the really sort of no-brainer thing here to do is to now apply machine learning and really start to try to relate what's going on here. And, you know, there's a lot of debate over what these patterns are. So this is sort of a new area of publications coming out from a bunch of different labs now. Some people are really in here seeing the behavior, right? They're like, I'm sure if we just could understand every little thing that the mouse is feeling and doing, we would see a representation of that in this cortical behavior. And that might be true. But there's a lot of other people who are thinking about things like replay, right? About the way that if an animal is now thinking about how it was running a few minutes ago, you would expect there to be some cortical representation of that. So how on earth do we disentangle that from the ongoing stuff that they're actually doing at the moment? And me, personally, I kind of like the idea that there's some background music in there as well. And I don't exactly know how to sort of explain and interpret that, but I want to relate this to the resting state fMRI experience, which is when you're sitting head fixed in a magnet and all this, you know, thoughts are going through your mind and your brain is generating all of these patterns. And honestly, I don't know if I particularly care whether it's all interpretable or not interpretable, but this background activity has to have some impact on how you respond to an input at any particular moment in time. I don't even know whether some people are saying, well, these little movements that we make are represented in the cortex, but you could also argue that these patterns in the cortex might be causing us to make these little movements that we tend to make, right? So there's always this chicken and egg. So it's very complicated. If anybody has some good ideas about this, I'm very much open to discussing kind of all the different things we can do. Right now, we're also going to be sharing a big cohort of this data. But very quickly, since I have no time left, one thing to note is that here I'm just clustering this data, right? But what I'm not showing you is the way this data is correlated to itself. And that's really relevant for this question of functional connectivity. Each of these regions has this different time course, and all these time courses are different and interesting. So I can actually do a correlation matrix between those regions and now actually start to understand how those regions are actually correlated to each other. And so that data actually can be represented as a movie here where each of these regions is now showing where it's correlated to. And now you're seeing what could be intuited as these functional networks, right? And so they're revealing all these patterns and regions of the brain, like I said, that don't exist on the atlases, but they're kind of cool. But then you can look at the dynamics from one moment to the next, and they seem like they change. And this is what brings me back to my worm. So they're all different. So now what we can think about doing is actually doing those correlation coefficients over different time windows and looking at the way that those different kind of sequences and patterns might be dynamically changing over time. And what I did was I just generally generated all of these different correlation coefficients and then clustered those and sort of salaciously called them states. And so now you could sort of imagine that the animal's kind of switching between these different patterns of functional connectivity just in the same way as the worm that I showed you before. And so we've been doing this now on a much larger scale, looking at the animal over the course of 80 minutes and looking at whether we can kind of create these clustered sort of dynamic states and look at when they occur in relation to the behavior. And particularly, I like this orange state, number 4, which seems to be sort of what happens to the mouse like when it's not running, when it's kind of like zoning out. And I'm wondering whether this is somehow a depiction, and again, I like my analogy to my background music, because it's kind of like what's ticking away in my head at that particular moment in time. And as a sort of spoiler alert, this kind of analysis is actually what the resting state fMRI guys are doing now. They've noticed this too, that in the course of their runs, they don't just see one functional connectivity, but they see it changing over time. And they're actually finding that those switches between those states seem to be indicative of disease states. And so I sort of have this romantic view that these kind of, your ability to switch between these dynamic states might be a representation of sort of your brain's capacity to tackle change, for example, which could be quite an interesting marker for brain disorders or psychiatric disorders.